Good morning, students. Can you all hear me okay? Good morning, students. Can you all hear me okay? Okay, so I'm going to get started. So today we're going to talk about the second part of DNA replication. Um, Last time we talked about leading and lagging strands and how. Can you all hear me okay? How the polymerase and actually how the 7 proteins supposed to do its job to help to replicate DNA. I'm going to get started. So today we're going to talk about the second part of DNA replication. Wait, I need to turn something off first. Last time we talked about leading and lagging strands and how. Can you all hear me okay? I was wondering why I have echo because I have another window open. Okay, so now we are ready. So if you remember, in order for DNA polymerase to work, it will require an existing 3' end. So in another word, it needs another nucleotide in front of it. So then there's a 3' hydroxyl group, so the polymerase can recognize and add the nucleotide to that end, correct? Now think about it. If polymerase always require primer, eventually when you are in the lagging strand, so here's the picture showing you on the bottom. When you get to the end of the lagging strand, and when you remove this last primer, which is the green showing up here as RNA primer, then you won't be able to fill this gap because there's no existing 3' end for the polymerase to work on. He can add on to the pink, which is the 5' end. Can you see that? So there's always going to be a gap at the end of the lagging strand. And the problem about a gap like this is that you will have a single strand DNA. Remember, DNA is always double strand. But in this case, you have a single strand DNA. And what happened in your cell is that when they see a single strand DNA, a lot of times they recognize it as a virus or viral DNA. So then they go, well, it's not normal. So they would degrade or digest the single strand DNA. So as a result, when chromosome replicate each time during cell cycle, so let's say your cell divide 10 times. So each time when it divide, a little bit of that end pieces of the chromosome will get degraded. So then your chromosome will get shorter and shorter, and you can't do that, right? Because you have genes that you have need to be there with the complete sequence to make proteins. So then how do we deal with it? Obviously, we survive. So what do we do? So the end of each chromosomes, they're called telomeres. So telomeres actually don't contain any genes. They're kind of extra DNA for this particular purpose because the cell or the body knows that the end of the chromosome is going to get degraded. So they have extra at the end just for that purpose. Okay, so like I said, the purpose of the telomeres, the end of the chromosome, is exactly for this purpose. It doesn't contain any gene sequence or information to make a protein, and it's okay to get degraded. So you get extra pieces there. So this picture here is showing you the little bright dot is the end of chromosome. So Aneri is asking, those are 7 proteins. Remember, proteins can be enzymes. So the 7 proteins we talked about last time for DNA replication, some of them are enzymes like helicase, DNA polymerase ligase, right? But some of them just some of them are just proteins, like single-strand binding proteins. So in somatic cells, if you remember, somatic cells are the cells throughout your body, like liver cells, skin cells, except sex cells. So somatic cells is always diploid, has 2 copies of each chromosome, each type of chromosome. So for each type of chromosome in somatic cells, you have your father's paternal, and then you have your mother's maternal. That's why I have 2 copies, and they look very similar, but they're not identical. And remember, I said that they don't touch at all. They don't touch at the centromere. Now, so what the slide is talking about is somatic cells don't have any telomeres. No, sorry. Somatic cells don't have any telomerase. Okay, and we're going to talk about telomerase in a minute. Now, remember telomerase, look at that word, it ends with ASE. So that means this is the enzyme that work on telomeres. So in somatic cells, every time the cell divides, the chromosome will get shorter and shorter. And eventually, when the chromosome gets so short that all the telomeres are gone, it would stop cell cycle, so it won't divide anymore. Okay, so when the scientists or the biologists saw this, they hypothesized that each cell has a certain lifespan. So it can, let's say, each cell can only divide 10 times. How does it know it already divided 10 times? The only way it knows is by knowing the length of its own telomeres, because if the telomeres get really short, then the cells will know, well, I might need to stop dividing because that's already too short. If I divide again, then I don't have any more telomeres left. So biological aging, or biological clock, as you would say, scientists think that it's based on the length of telomeres. So let's talk about telomerase. So it's the enzyme, right? So as the telomeres get shorter and shorter through each cell cycle, telomerase is the enzyme that actually, in a sense, remake the telomeres. So as it gets shorter, it's making it longer again. But the catch is this: telomerase only exists in sex cells. So somatic cells don't have any telomerase, only sex cells have. And why is that important? Because remember, your sperm and your egg, they need to have the longest possible telomeres, so you can give that to your kids. You don't want your kids to start out with really short telomeres. Now, this is you should have watched the video for this already. It's talking about how telomerase work. And this is just a picture showing what that video says. So telomerase is an enzyme, which is the green little cloud thing here. And the thing is, it contains its own stretch of RNA template. So if you see that on the picture here, it shows that it will use the RNA template to extend the end of chromosome, like that in the red. Can you see that? So that's what the job of telomerase are. They extend the end of chromosome, so you get longer telomeres, so the cells can keep dividing. Okay, do we have any question about telomerase and telomeres before we go on? Okay, now, since telomerase is the enzyme that lengthens the telomeres so the cells can keep dividing, right? So then think about it. That could be a really good way to extend life, right? So the scientists think that, oh, they find the fountain of youth if we can put telomerase into the somatic cells, then the somatic cells like your liver or skin can divide forever. Then you won't have to die, right? The cells don't have to die. So they actually tried this experiment in mice. Um, they put telomerase back into the somatic cells in mice, and they should actually be able to extend the lifespan of the animal by 24%. So let's say if you get to live to 100-year-old, so by doing this experiment, you can live to 125 years old. But there's always a catch, right? It turned out that animal that get this kind of dose of telomerase in the somatic cells, since if the somatic cells can keep dividing, not stopping, does that remind you of anything? Cancer, right? So it's a very fine line. You can live a lot longer, but the chances of getting cancer is also really high because the cell keep dividing, and as it get older, the checkpoint tends to fail. So he's always a catch for anything good. Okay, B is correct. So I just open up the worksheet. This question right here will help you answer question number 2 in the worksheet. So let's take a look. Can you see question number 2 say, why can't DNA polymerase add nucleotide to the end of the lagging strand? And this question, you can find the answer in the question you just answered. Okay, let's go to the next one. Next question. Yeah, yeah, Misty, what's the question? Yep, B is correct. Now let's look at some of the facts about replication. We talk about how DNA needs to be replicated during the S phase in interphase, and S phase only have anytime. I think its time is like 7 to 9 hours. So within 7 to 9 hours, the polymerase need to be able to add close to a billion nucleotides. So if you do the math, they gotta figure out that polymerase actually add about 1,000 nucleotide per second. Think about typing. Can you guys type 1,000 alphabets per second? Probably not, right? So according to this, they say the error rate should be 1 in 1 million base. So every time you replicate DNA, you should get approximately 1,000 mutations. But we don't get that many, and how does that work? So let's look at Miss T's question first. So the primer is added by polymerase 3. No, remember primer, polymerase 3, DNA polymerase 3 cannot add primers. Primers are added by primase. Last time we talked about it, right? So in this scenario, let me see if I can open up that picture again for you. So in this scenario, what happened is the primer, if you look at the yellow here, this is the RNA template that carried by telomerase. So using that template, they can extend that lagging strand right there, the gray, the white, so making it longer, right? And then so the primase can add new primer on top. So primer is not added by DNA polymerase at all. So by extending the lagging strand longer, then there'll be room to add primer by the primase, and so then they can fill in again the end of the DNA. But you're right, at the very end, like this picture here, at the very end here, there's still a little bit of the DNA will get degraded because you can't fill it all the way to the end. But that's okay because you already extended the telomeres. So you get it really long now, and if you cut it up a little bit, if you cut up the end a little bit, that won't affect it. Okay, let's try to go back. Now, so what happened in, remember we were talking about you should have at least 1,000 mistakes every time you replicate your DNA, because just like think about typing, when you type, you can have typo, right? And that exact analogy work out because as DNA polymerase 3 add nucleotide, right? Typing each nucleotide one at a time, it can also proofread its own work. Kind of like you, when you type something and you think, oh, you see that you have a typo, you go back, delete, and add the right thing. That's what DNA polymerase 3 can do. There's a particular part or unit in the polymerase 3 that is called epsilon that can actually proofread its own work. So the way it does it is it has an exonuclease. So nuclease means that it cut out DNA. So the epsilon unit, as you see on the bottom part of this picture, can cut out the blue nucleotide and put the right one in. So the proofreading function of DNA polymerase 3 is done by the epsilon subunit. So that will increase its accuracy that will reduce its error rate by 1 times 10 to the minus 7. Okay, so let's say after DNA have been replicated and the proofreading function is going on, but then there's still some mistake left after replication. So there are constantly proteins within the cell to scan all the DNA. So look at this mismatch repair picture here. You can see that there's a mistake right here, that the shape just looks wrong. So when the protein comes, and then the light green on the back here, the protein come along and scan it and go, oh, this is a mistake. So there are 3 steps to fix a mistake like this. So it's called mismatch repair. Mismatch means that the nucleotides mismatch. A is not matching with T, G is not matching with C, right? So they repair it by number 1, cut out the bad part. So nuclease supposed to cut out the bad match right there. Number 2, DNA polymerase 1 can replace, after you cut out the bad part, you have a gap in between, right? So then the DNA polymerase 1 can come in and fill in the gap. So the light blue, that's what happened. And then there's and then ligase, the enzyme comes in and stitch the fragments back together. So it's kind of like surgery, right? They cut you open, remove all the bad part, and then they stitch you back together. So that's what mismatch repair is. Now, this what we just talked about. If you look at the worksheet, question number 4. If you see question number 4, the first question here is, what is this a diagram of? This is a diagram of mismatch repair, in case you can't see it very well. Okay, so the 3-step ABC is the 3-step we just talked about, how you have the nuclease for A, you have the nuclease to remove the damaged DNA first. And then second step is you have the DNA polymerase 1 to fill in the gap, which is right here, right? And then the last step is that ligase will come in and stitch the fragments together. Yep, the answer is D, it have to have existing nucleotide to add on to, right? So it cannot initiate or start a strand. Now, besides mismatch repair, we're only going to talk about 3 kinds of repair, right? On your worksheet, on question number 3, it asks about the 3 types. So you have to write down DNA polymerase can proofread itself, proofread its own work. Number 2 is mismatch repair. Number 3 is this one that I'm going to talk about. So if you expose DNA to UV light, sunlight, what happens is the thymine, which is the T here, when it's exposed to UV light, it can cross-link. That means it can stick together, okay? And so when it stick together, it's called thymine dimer because you have thymine becoming 2 things, right? So to fix this kind of mistake, that means the DNA were fine. After you replicate it, the matches, see A still match with T, there's not a mistake. It match a complementary base pair. And the mistake is because you expose the DNA to UV light, and now the thymine, the blue here, they cross-link here. That's why it looks funny. So this is also a mistake because structurally it doesn't look right. So to repair this, you have to have nucleotide excision repair, okay? So it's the same steps. First of all, the body will be able to detect the mistake, right? And then the nuclease come in to remove the bad DNA, leaving a gap. So nuclease come in, so it's the same 3 steps. And then the DNA polymerase 1 come in to fill in the gap with the correct nucleotide. Okay, and now the last step is the ligase will come in to stitch this fragment to the rest of the DNA. Okay, so this is the 3rd repair mechanism we talk about. So there are only 3 types. DNA polymerase can proofread its own work. Number 2 is mismatch repair. Number 3 is nucleotide excision repair. Now here is an example of what happened if your DNA repair mechanism is not working. So XP is a genetic disease that the individual don't have a functional nucleotide excision repair system. So when these people go outside in the sun exposed to the UV light, they can't repair the DNA damage caused by UV light, the thymine dimer we just talked about. And so when they go out in the sun, they will have like blister and sunburn really, really bad. Now for this concept map here, because you guys cannot use my smartboard, so let's just do it this way. I see some of your name here. I'm gonna call on you to give me the answer, okay? Or to work on whatever answer is. So let's do Misty, can you do the first bubble here, say proofreading? Saul, you do this bubble, say cutout damage. And then Brianna, you do this bubble, say prevents chromosome shortening. Harley, you can do this one, say damage. Who else do I have? And Neri, you can do this bubble, say untwist strands. Heath, you can do this bubble, say relieves torque or tension. Relief, that's what it's supposed to say. Samantha, can you do this bubble, says holds open? Who else do I miss? Who else I didn't call? I forgot already. Oh, Dahlia. Dahlia, can you do this bubble? Say head start for enzyme. I think I don't have enough people here. Oh, Sarah. Sarah, will you be able to do one? Sarah, can you do read a continuously assembly? I don't remember if I did I call you at did I give one to in already? Oh, okay. David's and in. Okay, in, why don't you do this continuous assembly? David's, you can do fragment here. You guys are doing a good job. It's working out. Okay, let's wait for, okay, Davis, type in Sam. Sarah, are you able to do it? Yeah, yes. Okay, let's see what you guys have is correct. Okay, so Misty got proofreading here is DNA polymerase, correct? And then cutout damage, what do you guys, who has cutout, oh, cutout damage is here. Cutout damage is nuclease, exonuclease, which is B. Okay, damage hardly has mutation, which is good. So damage here is mutation. Untwist strands here is helicase A, which is correct. So heat has relieved tension is topoisomerase, which is L, correct. What holds open the double strand is going to be single strand binding protein, right? So we're up on this. Prevents chromosome shortening. Brianna has it is telomerase, which is H. And then reads and copies DNA. Once again, this is DNA polymerase. Okay. And here is head start for enzyme. This will be what? RNA primer, because RNA primer is what is needed for the enzyme DNA polymerase to start on. Okay, so we're here. Recent start copies DNA is polymerase. This one is RNA primer. Continuously assembly is leading strand, which is M. This continuous assembly is lagging strand, which is G. Fragments, you guys got it right, is Okazaki. What links this fragment together is what? Ligase. Good job, good job. Now remember, we talk about central dogma. Basically, the central dogma for molecular biology is about how the information flow, right? It all starts with DNA, and then information get copied into messenger RNA, and then this information is taken outside the nucleus into the cytoplasm, where ribosomes will read the messenger RNA information and create a protein according to that information. So you should remember this by now. From DNA to RNA is called transcription, which is inside the nucleus. From messenger RNA to protein is called translation and is outside the nucleus. Good, that's correct. So just a reminder. Now you should know these terms too, in case you have forgotten. So I'm not going to go into detail. And the key thing here is to remember the primary structure of protein, which is the sequence of amino acid in the protein, is determined by the sequence of nucleotide sequence in DNA, right? That's what we learn about transcription and translation. And the primary structure, which is the sequence of amino acids in protein, determines the shape and function of that protein. Yeah, Misty, you're right. When they say hypothesis, they have some evidence that is doing, is going through Okazaki fragment and this continuous lagging strand, but they don't really have a solid proof. It's not like gravity. You can actually see it work. So it can change anytime. Who knows? Come on, let's vote for this question quickly. Should be easy one. Yep, the answer is A. Just remember DNA contains information to make a protein. So I talked about this already, how the order of nucleotide determine the order of amino acid sequence, and that's what makes a protein. One thing before I forget, now um, from this worksheet on, if it's possible, I want you to download the worksheet from D2L as a Word file. And then you can use your computer to fill it out and then email it to me, because some of your pictures are really hard to see with the glare, stuff like this. And sometimes the picture is sideways. I have to, it's just hard to see if it's possible. And if you don't have a Microsoft Word on your computer, you can always go to eWolf and use the Microsoft Office Online from eWolf. So they have an online Word that you can use. So just open it up there, save it to computer, and then that way it's easier. If you don't know how to download it, you just click on the file and then you scroll to the bottom of the window after the file open and click, there's a little button say download. Okay, so here is talking about genotypes and phenotypes. You will know these two words later on even more in detail when we talk about genetics. So genotype is just like what it means is your genes. What type of genes do you have in your DNA? So determined by your DNA sequence. Phenotype is talking about the physical characteristic you can actually see, the color of your hair, your skin color, your height, and stuff like this. And all the physical characteristic that we can see is made of proteins, right? Your hair color, your eye color, your height, whatever it is. So DNA sequence, genotype, which you cannot see, determine phenotype, which is a physical characteristic you can see, because DNA sequence determine the amino acid sequence, which is in the protein, remember? Now here is showing you that if you have mutation or just one nucleotide change in the DNA sequence, so the top red here, you see that it says genotype. You suppose you see that there's only one difference. I think this picture got shifted a little bit, but what they're trying to tell you is that there's one difference in nucleotide as far as the DNA is concerned. Okay, and because of that, if you go to the bottom, say protein, you see that it changed a protein, one amino acid. So one nucleotide change can change amino acid, can change the protein amino acid sequence. So because of this protein sequence differences, you can see that the mouse can be brown or can be off-white. So this slide is trying to show you how the DNA sequence determine the amino acid sequence in green, which then determine what you see as physical characteristic. Now here, mutation, you see here there are a lot in different things. Mutation here in particular is referring to a permanent change in the DNA sequence in the hard drive that gives you instruction to make a protein, right? There are a lot of things can cause mutation. For example, you can have, like when we talk about UV light, you will go out in the sun. That's why they said that you need to cover up with sunscreen. It can cause skin cancer because UV light can mutate or cause damage to your DNA. And there are two kinds of mutation we're going to cover today. One is point mutation, and that just involves a single nucleotide change. Very small scale, usually affect only one gene. And then, of course, you can have a massive chromosome level mutation where a chunk of the chromosome is damaged. So that will involve many genes and is in a large scale. So these are how mutation occur. You can expose to UV, like I said, radiation, or you can smoke. The chemicals in cigarette can also cause mutation in your DNA. Now, when we talk about point mutations, you're thinking about one nucleotide, okay? So there are a few ways you can do this. Think about it. If you have a stretch of sequence or DNA sequence, you can either replace one nucleotide with another. That could be a mistake, right? So that's kind of like the mismatch nucleotide thing we talk about, mismatch repair. Or you can delete one nucleotide by accident, or you can insert extra new nucleotide. So that's only the three scenario, right? That's possible when you deal with one new nucleotide. So let's talk about the three types of point mutations. And we'll take a break at 10:30. So the first type is called missense mutation. If you look at this picture, you see that the white here, the second amino acid, tryptophan, supposed to be TGG, right? If you have a substitution of the nucleotide from G to T, then you go from tryptophan to cysteine. Can you see that? Everything else stayed the same, but you are substituting one nucleotide to another by mistake. Maybe it's because the repair mechanism didn't pick it up, didn't see this mismatch and leave it there, and that could be an example. So missense mutation will cause change in a single amino acid because you have one nucleotide change. Can you see that? So that's missense mutation. Now, because the genetic code is conservative and redundant, remember that that means that the first two nucleotides is the same, and the last one can be A, C, G, T, any one of those, and that's redundant, right? You could have something called silent mutations. So if you look at this example on the slide here, you see that the first amino acid is supposed to be TAT, right? But if you change it to TAC, it's still okay. It still gives you tyrosine. It doesn't change amino acid at all. Because if you look at your genetic code table, you can tell both TAT and TAC will give you tyrosine because the last nucleotide is flexible in pairing. Remember that wobble hypothesis we talked about? So that's what I mean by genetic codes, because genetic codes are conservative and redundant. That's why you're allowing silent mutation, which is a good thing because that means that not every single mutation will cause a problem, in amino acid sequence. So a lot of times mutation is overlooked because of this silent mutation thing. Now the third type of mutation is nonsense mutation. Okay. Once again, like the other two we just talked about, is a base substitution or is a nucleotide substitution. Okay, if you look at this example here, look at the second amino acid here. On the top is TGG, supposed to code for tryptophan, right? But if you have a mutation to change it to TGA, now TGA happen to be a stop. So you can see that this protein will be stopped prematurely. So it depends on where the stop mutation or the silent mutation is. If it is in the very beginning of the protein sequence, then you have a protein that's too short, that's not functional. But sometimes if this stop or this silent nonsense mutation is toward the end of the protein, it might be okay. So it just depends on where this nonsense mutation happens to be in. Now let's do a quick review. So we talk about 3 mutation, right? We have, and these 3 mutation, I'm going to the worksheet right now, are all caused by base substitution. That means you're replacing one nucleotide with another one. And the 3 kinds of point mutation that are caused by nucleotide or base substitution is what? Missense mutation, right? Nonsense mutation, and what is the other one? So you have nonsense mutation, you have silent mutation, which doesn't cause any change, and then you have missense mutation. Those are the 3 caused by nucleotide substitution. Now we're going to talk about what happens when you delete or insert a nucleotide. Good. Now remember the last 3 we talk about, you're just replacing one nucleotide with another. You're not deleting or inserting, right? But this type of mutation, what we're going to go into now, is talking about what happens when you insert or delete a nucleotide. So here's an example, and this kind of mutation is called frame shift because you're shifting the whole reading frame. If you don't remember what reading frames is, here's an example. Now, here's a sentence, say, the fat cat ate the rat. This is a reading frame showing you a codon, 3 letters at a time, right? And you have to read it knowing where to start, where to end. Now, if you insert a letter in this sentence, you see the red here, and then everything gets to shift because it has to be 3 letters to make a word. So you see how when I insert a letter here, you don't get the same message anymore, right? This is what frame shift mutation is. Imagine this sentence is instruction for making a protein. It's codons, right? 3 letters, and each codon will give you amino acids. So then if you insert or delete a nucleotide, the whole reading frame will be shifted. So the example on the left here showing you this is the amino acid sequence right here, right? If you insert the white letter C here, you see that how the insertion will cause amino acid change after the insertion. So this is actually pretty bad. All the other 3 mutation we talk about, you only change 1 amino acid. But in frame shift mutation, you change many amino acid right after the insertion. Can you see that? So here's another example. So this will happen if you insert an extra nucleotide or when you delete an extra one. Yeah, so Anne is asking, I don't know for sure how do they get inserted or deleted, but I think sometimes the repair mechanism didn't do its work. So insert extra one, and sometimes like you said, it could be replication that it just put an extra one in there. So we don't know for sure. So here this in this slide is showing you frame shift mutation. You can insert 1 or 2 nucleotide and will shift the whole reading frame after the insertion, because remember codon is 3 nucleotide, is triplet. But here is the example here. You can have a non-frame shift mutation if you insert or delete 3 nucleotide at a time, because it has to be multiples of 3, right? So if you look at this example here, if you delete or if you insert these 3, actually everything else behind will be the same. You just have 1 extra amino acid. So the same thing goes with if you delete 3 nucleotide at a time, you're just missing 1 amino acid. Everything else will stay the same. So what is this kind of mutation called? Good, it's E, but what is the name of this mutation? Perfect, perfect. It's silent mutation where you can't see, right? Silent. Let's try this question now. Okay, so most of you vote for B, which is incorrect. Look at this. Because the first two codon is the same. So the first two amino acid will be the same. So the answer is C, where after the deletion, after the red T, everything afterward will be different. Remember that? Everything before will stay the same. Right? You remove this T, you get ACA, so the codon, the reading frame will all shifted after the deletion here. So let's look at the worksheet again. And then we'll take a break. So we just talked about the three types, and then here's this question. So what kind of mutation is caused by base insertion or deletion? We just talked about it. Okay, what kind? What kind of mutation is caused by base insertion or deletion? Yes, frame shift. So in here you have to write frame shift, right? This one you write frame shift, and then you'll be able to answer question number 8 here. What kind is worse? Is it substitution worse or insertion or deletion worse? And you'll be able to tell me why, right? Now here, I already mentioned what characteristic of genetic code allows silent mutation. And you should be able to answer that too. You know how it is what? Redundant, conservative. Now, so the rest of this worksheet is on translating RNA. So this is RNA. How do you know it's RNA? Because it has uracil. So you need your genetic code table to translate this stretch of RNA into amino acid sequence. So here asking you to translate, right? So you write your amino acid sequence on the bottom here. So this worksheet, you should download it as a Word file and just type your answer in and then email it to me, because there's no pictures that you need to draw. Okay. Now, when you translate a stretch of RNA, what do? Do you start with the first nucleotide, or what do you do? Do you remember? Good people, very good. You still remember. So number one, use here. You scan for the first AUG, which is this one right here, right? And then you start with this AUG, and then the next three, and then the next three, go on and on. So good. So let's take a break for 5 minutes, and we come back and finishing it up. It shouldn't be that long. So look over your worksheet, see if you have a question. 5 minutes break. Plenty of time. Okay, let's come back now. I think it's been 5 minutes. So this is the last 2 slides for this, and then we're going to go on and talk about the lab, right? Today, supposedly on schedule is the lab 10. So come back now, come back now. What? Okay, so we just talked about point mutation, where you change one nucleotide, you insert one, delete one, stuff like this. So what about chromosomes? What if you have massive amount of mutation? Okay, so when we talk about chromosome level mutation, what we're looking at is there's two conditions here. One is called polyploidy, means that, remember how everybody has two sets of each type of chromosome, two copies, mom and dad. In this scenario, you have more than two copies of each type. So the example is like instead of humans should have 46 chromosomes, right? If we are polyploidy, we could have 3 times 23, like some 69 and stuff like this. So they're talking about more than two sets of chromosomes. No, the next one is called aneuploidy, which is this abnormal number of one type of chromosome. And the example here is trisomy 21. So this is chromosome number 21. Trisomy means that instead of two copies of 21, you have three copies. And this is commonly known as Down syndrome. Now here are the pictures. So you could have chromosome deletion where a chunk of chromosome just get deleted out, so it gets shorter, right? Simple enough. Chromosome duplication is where an area of chromosome somehow get duplicated, so it get longer. And chromosome inversion is a segment of chromosome get cut out and then flip upside down and insert back in. So the length of the chromosome didn't change, but the the segment of chromosome is the order is reversed in a sense. Insertion is between two chromosomes. As you see, the yellow one, part of the yellow one get cut out and insert into the smaller gray one. Okay, so that's what insertion is. Translocation is that you're exchanging parts. So in insertion, you're not exchanging anything. The yellow one just insert into the gray one, right? In translocation, you have exchanging parts. So the green and the red exchange. So they're kind of like a hybrid, right? They're part of the green now is red, part of the red now is green. So that's what translocation is. And I think this is the last one. But about chromosome translocation, there's a question on the worksheet, the very end. They asked you for a chromosome translocation. So that picture I just show you how the chromosome exchange parts. And inversion is the part of the chromosome get cut out and then you flip it upside down and put it back in. So you just use that picture to answer these questions, okay? Now I'm going to go into the lab. I hope that you guys already done the virtual PCR and watched the PCR pre-class assignment, all the video. Now, before I go on talking about the lab, is there any question about DNA mutation? Ooh. Okay, so let's talk about PCR. PCR stands for, I think I have another slide, but PCR is a technique that is used to profile DNA. So remember in the old days, when they want to take your fingerprint and each fingerprint is very specific or unique to individuals. So nowadays, besides fingerprints, they also look at your DNA because your DNA profile is unique for each person. And the way to look at your DNA is through a technique called PCR. And since you don't get to do the lab, so we're going to just talk about it and show you some pictures of it. And you will still be able to answer the lab questions, except the part about asking you who the suspect is. For that question, you just leave it blank. You don't have to. So I already talked about DNA profiling is looking at your genotype, your particular genotype. Okay, so if you have watched CSI, I know that all of you have. You always see the policeman collecting samples at the crime scene, right? And some of these samples they collect will contain DNA. Just remember, where do you find DNA? You find DNA in the nucleus of the cell, correct? So the samples that they collect has to have nucleus. So here are some samples like blood, semen, urine, hair. Now hair is a very tricky thing. The hair they collect has to contain a follicle at the bottom where the cells are. If you just collect a little bit of hair at the end, doesn't have the follicle, then it's just proteins. There's no nucleus, right? Protein is just proteins. So for hair, they have to have the follicle at the bottom. Now, so when you guys go commit a crime, make sure that you don't leave any of these samples at the crime scene. So the little sample that I collect that contain the nucleus and the DNA will be extracted. And then the DNA, after they extract the DNA from the crime scene sample, the amount is very, very tiny. Okay. And so that's why they need to do PCR on it. Now, but what part of the DNA do they look at? They don't look at all the area. They only look at a certain area called short tandem repeats, and they are anonymous because these short tandem repeats, STR, actually don't contain any information for making a protein. So if you remember when we talk about RNA processing, they're intron and exon, right? So they're looking at, in this short tandem repeat, most of the time is in the intron, where it don't contain any information for the amino acid sequence. Okay. Now, here's an example. So in intron, for example, looking at introns, a particular repeats called THO1 locus. So it's a locus means location. So THO1 location in the chromosome. The repeats that they're looking at is TCAT. Now, the reason it's called short tandem repeat. Number one is short. So you're looking at 4 nucleotides. Tandem means that they repeat each other. If you look at this stretch of DNA, you see the red repeats. So in this particular individual, there are 6 TCAT repeats. And because each individual has 2 copies of this chromosome, right? Mom and dad. So on moms, it could be 6 copies of TCAT, and on dads, it could be 8. So there are 2 numbers for each of these kind of repeats. Now here's an example. Mrs. Smith, look at the THO1 location of her chromosome. She has maternal chromosome and paternal. That's what I mean, right? In her maternal chromosome, the TCAT repeat could be 6, and paternal here could be 13. So her number for THO1 locus repeats is 6 and 13. Now, if they're only looking at one locus, THO1, then it could be there will be high possibility of one person being the same with the other, right? But FBI or the crime scene people don't look at just one location of the chromosome. So as now, they're looking at 13 different locations. So this is a picture of all the chromosomes you have. Remember, you have 1 to 22, and then the last pair is X and Y. So look at all these red boxes. They're looking at all these locations in different chromosomes, okay? And each one of this location of the chromosome will have repeats number that's different between maternal and paternal. So each one of these locations will have 2 numbers, like the THO1, right? The one I showed you before is 6 and 13. For this one, TPOX, it could be 1 and 3. So for each of these locations, we have 2 numbers represent the location, right? So there are 13 of them. So for all 13 to match between 2 individuals is almost impossible. Even identical twins won't match exactly on all 13. Now remember, because identical twins look identical physically, so it's all exons, right, where you express the protein information. But within introns, identical twins could be a little bit different too. So after they, the way the FBI look at the STR, the short tandem repeat, is by running it through a gel, and you guys will be able to see this later on. And they will look at, I'll show you one more in a minute. So they will look at a stretch. They run so each locus has 2 numbers. Remember I told you about? So it will show up as 2 different line on the gel. Okay, so this segment here is showing you the individual. They're looking at 1, 2, 3, 4, 5, 6, 7 different locus here. Now let's go back to PCR. So you find the crime scene DNA, which is tiny bit in the crime scene, right? And then you want to look at the crime scene DNA's STR region. And the only way to do it is to amplify the tiny bit of DNA in the crime scene through a technique called PCR. Now, PCR stands for polymerase chain reaction. You know what polymerase are, right? It's DNA polymerase. So this is doing DNA replication in a test tube, okay, by using polymerase. And it's a chain reaction, and I'll show you why. And it amplifies the tiny bit of DNA from crime scene into massive amounts so you can actually analyze it and do a profiling. So you guys should at home listen to this PCR song, very fun to listen to, and then see if you can identify the answers for these questions. So to have the PCR reaction, which is DNA replication in a test tube, you need a few components. Number one, you need the crime scene DNA, right? That needs to be amplified. Number two, you need the raw material, the DNA nucleotides, the ACGT. Number three, you need the polymerase, which will add the ACGT to make DNA polymer. Number four is primers. Remember, polymerase do not work without a primer because it requires existing 3' end. So we're going to add primers to the reaction so polymerase have something to add on to. And then after we mix all these reagents into the tube, we put the tube into a thermocycler. Basically, it's a machine that can fluctuate between different temperatures. Now, if you remember, polymerase is an enzyme, correct? Enzymes is from protein. And if you remember from your protein chapter, protein get degraded or denature or unfold in high temperature, correct? So PCR has to go through a particular step that using really high temperature. Without and the polymerase that they use in PCR is a very special polymerase. Scientists identify it from Yellowstone those hot spring. The temperature gets really hot. They find bacteria in there. That's what make the hot spring has different color. Remember that? If you ever seen pictures of the Yellowstone hot spring, those bacteria in there are able to multiply and divide. It's because they have a certain kind of polymerase that can withstand the high temperature without denaturing. So then scientists look at those bacteria and isolate the gene for that polymerase in particular, and then they can use it in the PCR. So the 3 steps in PCR is number 1, denaturing, annealing, and then extension, okay? So the denaturing step is 94 degrees Celsius, so it's almost boiling. And because remember the first step in DNA replication, you need helicase, right? But we are not putting in helicase here. And it turned out if you heat DNA to high enough temperature, it will unzip all the hydrogen bond between the nucleotides. Remember that? Because you're giving the DNA a lot of energy. So you're shaking it like vigorously, so those hydrogen bonds just kind of fall apart really naturally. So this is the step where the polymerase needs to be able to withstand because you have to heat up the DNA to open up the double helix. So then after you open up the double helix, you will lower the temperature to annealing. So what is annealing? Annealing is you are letting primers that you add to the reaction to anneal to the DNA. Okay, in that way, then the polymerase can work in the third step, extending. Extension means that you're making DNA polymer. So with the primer anneal in step 2, then polymerase can have the 3' hydroxyl group and add nucleotide to the primer. And then we repeat these 3 steps 35 times. So we'll go from, here's an example. If you repeat those 3 steps 35 times, what you're doing is you are taking, let's say initially you only have 10 molecules of DNA. If you repeat 35 times, you're bringing that to 35 exponent. So you can start with very tiny bit and end up with millions of copy of DNA. And that way, remember crime scene DNA is very little, and then you amplify it to millions of copies. That way you can use it to analyze. Okay, so let's answer Misty's question. It's actually a good one. Well, the few skin cells is not that much DNA. It depends on how much DNA they can extract from those few skin cells. If they extract enough, they will go through PCR and and get a lot more DNA from there. But sometimes the DNA left in crime scene can get degraded. So let's say you start with 10 skin cells and 5 of them not good, and you don't get any DNA out of it, and then there's only 5, and it's really hard. I mean, that little DNA, even PCR sometimes have a hard time. You need a minimum amount to amplify. If you only have if you have too little DNA, you can't really get a full picture. Now, so what is PCR good for? After you're amplifying the DNA and you're looking at specific region, like I said, you're looking at STR, right? Different STR and different chromosomes. So here is PCR can be used for looking at can be used for paternal tests. Okay, so if you look at this picture I have here, you can see they're looking at the TH01 locus, right? And for each individual, the mother and the father, you have two chromosomes for this particular locus, TH01. So you see that for the mother, her number is 3 and 6. And for the father, his number is 10 and 5. So once again, remember for each locus, each individual will have 2 different numbers. Okay, it could be the 2 same numbers, whatever it is, but we will have 2 numbers. Now, then if you look at the kids, remember for the children, one of their, for each chromosome, one of the copy has to come from the mother and the other copy has to come from the father. So you look at this picture, which one of this kid is questionable? Try it. Yep, you guys got it right. So just to be sure, look at child C. This is from the father, right? This one is from the mother. But if you look at child F, this one is from the mother, but this one doesn't belong to the father. Ooh, that's a problem. So that's how they use PCR to look at particular locus to identify whether the kid belongs to the father or the mother. Well, the mother is easy, right? You give birth to the kid, you have to be the mother. Well, I guess this day it doesn't have to be. Well, but anyway, this this is how the genetic testing works now. Now, PCR is also good for crime scene when you're looking at rape and stuff, right? If you look at this picture here, this is a gel electrophoresis. We're looking at the same thing. For each locus, we have 2 copies for each locus, right? So you look at the victim here. She has this 2 copies, and you have suspect 1, 2, and this is just the victim. The female cell is the victim. And then this is the crime scene sperm DNA. You see that? These 2 here. And then, of course, you have to have the boyfriend DNA here. Make sure it's not the boyfriend gone crazy, right? So the boyfriend DNA doesn't match the sperm DNA. So it's not the boyfriend. But the sperm DNA match whose DNA? Does it match suspect 1 or suspect 2? Yeah, you got it. See the sperm DNA from the crime scene or from the female victim matches suspect A at 1. Suspect 2, there's actually 2 numbers, but they kind of really close together, so they overlap. So you can see that there's supposed to be 2 in this thick one here. So are we good? So PCR is good to amplify DNA, to do a genetic profile, to look at sexual assault, to look at paternity tests and stuff like this. So we talked about this already. And this is the application. We already talked about crime scene investigation, genetic testing, and stuff like this. So in this lab, what you're supposed to do is I'm supposed to give you DNA from the crime scene and 4 suspect, A, B, C, D, and you do a PCR to look, just like the picture I showed you before, and look at which suspect matches the crime scene DNA. And the locus that you're going to look at is this particular locus called BXP007, 007. Okay, and so today, in theory, we're supposed to put all the reagent together in a tiny tube, let it run in the PCR machine, and then we'll run the gel next time. Okay, and on my schedule, next time is April 6th, so Monday. It's the time I'm going to show you how the gel works. Okay, so on Friday, we'll start a new chapter called meiosis. And then you also do the lab on meiosis too. It's a really short lab, it's activity lab. So this part is what we're gonna do on Monday. So we try to follow the same schedule we had before. So if you just keep an eye on it, it'll be okay. So I think I'm done with all the materials I want to cover today. Is there any question? Or does anybody know or not know how to download the worksheet from the D2L? If you don't know, can you just tell me now and I can walk you through it? We still have about 15 minutes left. So the lab report is not due until um, April 8th, next Wednesday. But you keep an eye on the post and pre-lab time, right? They should show you on the schedule. Let's just follow that. I know, actually, usually every semester I set up this PCR lab myself. That's one of the good ones. Quiz. Are you talking about quiz 4, Brianna? So what I'm gonna do, I know I posted saying that I will post the worksheet answers on D2L, but I think the better way to do it, because some of you didn't turn in your worksheet at all, I'll just email individuals who turn in their worksheet, and I'll just email you the answer if after you turn in your worksheet. I think that way is better. In case some of you got sick and have to turn in worksheet late, if I post on D2L, then they cannot turn in late. Brianna, for quiz 4, all the answers for quiz 4 is from the worksheet. So you should be able to find it in your worksheet. If you have questions, you can't find it, let me know. But like I said, all the questions is just either straight from the worksheet, from those chapters, or just a tiny modification. There's nothing really new. And you should be able to see your quiz 4 grade on D2L already. And most of you did really, really well. So remember, that is a review for the exam 4. So exam 4 will cover the exact same thing as quiz 4. And I was telling the others about exam 4. It's like half of it will be multiple choice and half of it will be essays. But not very long, but still, I encourage you to study, because if you have to look up everything, you won't have enough time. And the multiple choice or the questions in the exam will not be simply black and white. What is this? And like something you can just find it straight from the material. Is questions are mostly like, I already assume you know the material, so it's more like application questions, like if you know this, how will you use the information you know, kind of deal. So it's not just copy and paste kind of deal. Okay, I'm going to stay on until 11:20, just in case any of you have questions. If you don't, you're free to go. And I'll see you again on Friday, same time. So Harley asked what we do about the biofuels lab. So we'll still do it, but like I said, it's going to be virtually. So you go through well, I'm going to talk about it. When is the time? Let's see. The date I have it scheduled is, I think I'm going to talk about it during week 12, April 15. And so we're going to skip some of the actual lab, but the planning and design will be the same. But we plan to, well, we're just going to plan to do it virtually.